All right, so we're going deep into genetics today. Think of it like cracking open the instruction manual for life itself. We've got your notes and some textbook stuff on Gregor Mendel and his pea plants. Ready to go back to high school bio for a bit and see how those traits get passed down?
Oh, definitely. It's amazing how much one monk and his garden changed the way we think about inheritance. I mean, before Mendel, people thought traits just like blended together and kids, like mixing paint. But Mendel, he really shook things up with those pea plant experiments of his.
Yeah, those pea plants were his thing. Why do you think he went with them? I mean, was he just a big gardening fan?
Well, he was on to something. Pea plants, they're like perfect for this kind of stuff, grow fast, tons of offspring, and you can really control, how they pollinate. Plus, they have those really clear-cut traits like tall versus short or those purple versus white flowers, which makes it way easier to track how those traits get passed down, inherited.
So he's kind of like a genius then, huh? All right, let's get into one of his big discoveries. Dominant versus recessive traits. Can you break that down for us?
Okay, so imagine like a game of genetic tug of war, right? Dominant traits are kind of like the strong player always wins and shows up if they're there. Recessive traits are more like the weaker player. They only show up if they're paired up with another recessive trait.
So like a light switch. Dominant is the on position and recessive is off. You need two off switches to see that recessive trait.
Exactly. Great way to think about it. Mendel, he figured this out by crossing, purebred tall pea plants with purebred short ones. And that first generation, the what we call the F1 generation, they were all tall.
Well, hold on. So the shortness just like vanished. Poof.
Not really. It was more like hiding in the background. See, those tall F1 plants still carried the instruction for shortness, but the instructions for tallness were dominant. They kind of masked the shortness.
So it's like a secret code. Got it.
Yeah.
What happened when he crossed those F1 plants?
Now, this is where things get really interesting. In that second generation, the F2 generation, some of those short plants, they came back. It was about a 3.1 ratio. So for every 3 tall plants, you'd get 1 short one.
Whoa, hold on. What? So how do you explain that comeback?
Well, Mendel realized that each trait is determined by like a pair of factors, and we call those alleles now. We use letters for them. So a capital letter for the dominant allele and a lowercase for the recessive. We could use T, for instance, for tallness and T for shortness.
So the purebred tall plants had 2 T alleles, and the purebred short ones had 2 T alleles.
Yeah.
What about that first generation, those F1s that were all tall?
Right. So they got 1 T allele from the tall parent and 1 T from the short parent. That combination, that T, we call it heterozygous, which means, they have both alleles, but only the dominant one, the T, is expressed, so they're still tall.
Ah, so they had those shortness instructions hidden in their genetic makeup. Okay, that makes sense.
Yeah. And that brings us to Mendel's first law, the law of segregation. It basically says that during, when those gametes are forming, those sep cells, like sperm and egg, those allele pairs separate. So each parent, they only give 1 allele for each trait to their kid.
So it's like a genetic lottery. You get this random mix of your parents' alleles, half from mom and half from dad.
You got it. That's a great way to put it. That random segregation is what gives us that 3.1 ratio in the F2 generation. Some get 2 T alleles, some 2 need, and some get, 1 of each.
Okay, but how do we actually predict those ratios? Is there like a formula or something?
Well, that's where those Punnett Squares come in. They're like a visual tool to help us, map out all those possible combinations for the alleles and the offspring.
Ah, Punnett Squares, right. I remember those from back in the day. They look kind of like, tic-tac-toe.
Exactly. So picture a grid, and you write the possible alleles from one parent across the top, and then the other parent's alleles down the side.
Okay, so like for our tall and short pea plants, we have T and T across the top, and then T and T down the side.
Yeah, you got it. And then in each box of the grid, you just combine the allele from that row and column, like a little puzzle.
So we'd end up with boxes like T, 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 and T. That shows all the potential genotypes for like the kids.
Exactly. And then we can figure out the phenotypic ratio. So, like how many tall plants to short plants.
This is like making me feel like some kind of genetic detective. Okay, so Punnett squares are great for single traits, but what about like when you're looking at a bunch of traits at once? Did Mendel, did he ever do that?
He did, and that's when it gets even more interesting. He did what we call dihybrid crosses, where he tracked 2 traits at the same time.
2 traits at once. Whoa, that sounds Sounds pretty tricky.
It is a bit more complex, but those same basic principles still apply. So Zelda's looking at seed shape and seed color this time, round or wrinkled, and yellow or green.
Okay, so now he's got like 4 different possibilities. Did he start with those purebred plants again?
Yep, purebred for each trait, round yellow seeds and wrinkled green seeds. Cross those, and just like before, that F1 generation, they all had those dominant traits, round and yellow seeds.
So dominant still wins. But what about that second generation, that F2? Did those recessive traits make a comeback?
They did, but in all sorts of new combinations. He saw round yellow, round green, wrinkled yellow, A and D wrinkled green.
Whoa, so every combo showed up. Were the ratios still like that neat 3.1 split?
A little more complicated this time. He found a consistent ratio of 9.3.3.3.1. So for every 9 plants with round yellow seeds, you had 3 with round green, 3 with wrinkled yellow, and then just one with wrinkled green.
Okay, my brain's trying to picture how that all works out. Can we still visualize that with Punnett Square?
Absolutely, but the Punnett Square gets bigger. Now it's a 4x4 grid to fit all those allele combos from each parent.
Okay, I think I'm following. So we got to figure out all the different gametes, those sex cells that each parent could make. And since they're heterozygous for both traits,
you're on the right track. So each gamete gets one allele for each trait, right? Let's say R is for round seeds, R for wrinkled, Y is yellow, and Y is green. So our F1 plants, they'd be RRYY. So what gametes could they make?
Let me think. If those alleles segregate independently, like Mendel's law says, then we could have RY, RY, and RY.
Perfect. You got it. And then we take those 4 possible gametes from each parent and put them along the top and the side of our big 4x4 Punnett square. Then we combine them in each box just like we did before.
So 16 boxes, and that's showing every possible genotype in that F2 generation. No wonder there's so many variations.
You got it. And if you count up all those phenotypes in the 16 boxes, you'll see that classic 9.3.3.1 ratio that Mendel observed.
Punnett squares are pretty powerful tools. They really help break down these complicated inheritance patterns.
They do. And those dihybrid crosses, they led Mendel to his second law of inheritance, the law of independent assortment.
Another law. Okay, what's this one tell us?
Well, it says that when during gamete formation, those alleles for one trait, like seed shape, they segregate independently of the alleles for another trait, like seed color.
So it doesn't matter if a gamete gets the R allele for round seeds, it can still get either the Y or the Y for seed color. Totally on its own.
Exactly. It's like shuffling a deck of cards. You never know what combination you'll get.
So that's why we see all those different combinations in the F2 generation. It's like a genetic mix and match.
Exactly. And that independent assortment, it's one of the main reasons for all that diversity we see out in nature.
Okay, I'm really starting to get this. But does that independent assortment always hold true? Are there ever times when, like, genes don't inherit independently?
That's a great question, and it actually leads us to one of the limitations of Mendel's work. Remember, he was working with pea plants, and he just happened to pick traits that were located on different chromosomes.
So where those genes actually are matters.
Yeah. Genes that are close together on the same chromosome, they tend to get inherited together, so they don't assort independently.
Oh, so they're like best friends who always stick together.
Yeah, exactly. We call it linkage, and it's one of those fascinating things we've discovered since Mendel's time. But even though his laws have, some exceptions, they still give us that basic foundation for, understanding inheritance.
This is so cool. It's like peeling back the layers of a genetic onion. Always something new to discover.
You said it. So ready to take this beyond the pea patch and see how it applies to us humans.
All right. So we've covered pea plants like it's nobody's business.
Yeah.
But let's get real. How does all this Mendel stuff, apply to us humans? We're not exactly pea plants, and we can't really control who has kids with who.
You're right. We can't design human crosses like Mendel did, ethically speaking, but that doesn't mean we can't study human inheritance. We just got to be a bit more creative about it.
So how do we do that? Like, do Do we spy on families and track their traits?
Not exactly spying. We use pedigrees. They're like family trees that track, specific traits across generations, like a genetic genealogy project.
Oh, right. So we look for those patterns in families to see how traits are passed down. But how do we know which ones are dominant and which are recessive in humans? It's not like we can just look at someone and like see their genotype.
True, we can't see those hidden alleles, but we can make some guesses based on phenotypes, those observable traits. For example, if two parents don't have a certain trait, but their child does, that kind of suggests that the trait is recessive.
So it's like a genetic detective game. Are there any examples of human traits that, follow those classic Mendelian patterns?
Totally, tons. Like the ability to roll your tongue, that's a dominant trait.
Oh yeah, I can do that. Does that mean I'm definitely homozygous dominant for that tongue rolling gene?
Not necessarily. You could be homozygous dominant or heterozygous. Remember, you just need one copy of that dominant allele to show the trait.
Right, right. So I could have that recessive allele hiding out my genotype like a silent partner.
Exactly, and that's why those pedigrees are so helpful. By studying a trait over several generations, we can often piece together those genotypes.
So we've gone from Mendel's carefully planned pea plants to these like intricate human family trees. It's amazing how these simple principles he found are still so important today.
It is, and his work it's led to so many amazing advances in genetics and medicine. We can track genetic disorders now, understand what causes a lot of diseases, and even develop gene therapies.
It's mind-blowing to think it all started with a monk and his pea plants. What a legacy.
Totally. Mendel's work was truly groundbreaking, and his principles, they're still the foundation for understanding heredity and all that amazing diversity of life we see out in the world.
So to our listeners, remember that you're carrying a piece of Mendel's legacy in your own DNA. And next time you see a pea plant, give it a little nod. Thanks for helping us unlock those secrets of inheritance. Keep on exploring, everyone.